Okay, so today is our second lecture in the system biology module. So last time we talked about how we think about biology as a system. And today we're going to talk about one class of techniques that we are going to use to understand biology as a system. And that is to combine different types of omics data together. So in the class prior to this, we learn omics kind of one by one, each layer at a time. But today we're going to see how we can combine multiple layers together to analyze a biological system. So today's agenda, we're going to kind of went over what we learned about each omics and what information they tells us about the molecular state of the cells of the disease. And then we'll talk about the strategy that we can use to integrate multiple omics data type. And let's see some examples of how we interpret this information. Okay, so let's start with kind of what do omics data tell us, right? We learn about genomics, epigenomics, transcriptomics, proteomics. Right? What information, like what biological information do these platforms tell us? So we know that there are relationships across the omics layer. So usually when you join a lab, usually each lab will focus on a certain type of omics because it requires a different expertise, require different equipment, for example. But in order to understand how change in the genome, mutation, how does that disrupt the 3D organization of the chromosome, how does a mutation might disrupt gene regulation, how does a mutation might change the protein integrity, the function or the structure. We need to compare genomics data with other layers of information. And if we see change in epigenome, change in transcription factor binding, or change in histone modification, we want to know what it means. So we want to understand does those change in epigenetics, do they affect the gene expression? Do they result in the final change in protein activity, protein expression? And do change in protein affect the level of metabolite in the cells? So everything are connected. And change in one layer, like change in protein, can affect the transcription. Change in transcription of protein that modify the histone can affect the epigenomes. So changes can also go back and forth between layers. So what we will see next week in the next lecture is how we can draw the diagram, this kind of networks or graphical diagrams that connect components from the transcriptomic level, gene expression, to change in protein and metabolite levels. Next week we'll focus on how to draw these diagrams. But today we are going to just focusing on the conceptual of how we interpret transcriptomics data, proteomics data, and other omics data together to understand the mechanisms. So the main keyword for today is when we perform one omics, we notice the change in the cells, but we as a biologist, we want to know the explanation. We want to know the mechanisms. And in order to do that, we need to collaborate information across techniques. So for example, if we start with genomics, people like to do genomics because you can detect it easily. It's a static change in the DNA. There is no dynamics, no change in expression over time, no change across cell type. Genome is constant, so it's easy to study. But when you identify a mutation, you probably want to understand, to interpret if this mutation, if it occurs on the gene body, will it affect protein structure, will it affect protein function? We can predict using AI, but in order to make sure that this mutation actually cause the disease, you need data from proteomics layer to interpret that. And if the mutation occurs in regulatory region, like intron promoter enhancer, we want to know if this mutation change the regulation of gene of transcription, or does it change the splicing? In order to confirm this, you need to perform transcriptomics to see the actual change in gene expression, to see the actual switch of RNA isoforms, so that you can tell for sure that this mutation seems to be correlated with change in transcription. And if you want to know if change in mutation can cause a change in epigenetics, changing from closed to open chromatins, or does it affect the nucleosome or histone binding to the DNA? In order to confirm that, we need to compare genomics data with epigenetics data to confirm. If we identify copy number variation, we saw some genes being duplicated or being lost in the genome, we might have some hypothesis that if the copy number change, the gene expression level probably change, because more copies probably will lead to higher expression level. But to confirm that, you need to perform transcriptomics. And sometimes, when you have a copy number, the new gene copies doesn't need to be next to the old copies. Sometimes the new copies can be somewhere else in the chromosome. It can be attached to a new promoter. It can be attached to a new enhancer. So to understand the real effect of copy number variation, you need to compare the copy number variation result with the actual expression activity to understand if these actually change by the mutation. So basically, when you analyze genomics data, it tells you what change occur, what mutation occur. It tells you where the change occur, but it doesn't tell you the consequence of those changes. To understand the consequence of genomics, you need to perform different omics. So for example, we learn a lot about transcriptomics for gene expression. So when we see a change in gene expression, we know the consequence, there is some change in gene expression. As a biologist, we tend to ask the question, so what kind of regulatory change lead to this up or down regulation? Do we have more transcription factor binding? Do we have more open chromatin? What caused the up or down regulation? And we cannot tell that with just transcriptomics. We need to go back to genomics to identify if there is any mutation in the promoter, any mutation in the nearby enhancer. We need to go to epigenomics data to understand if the transcription factor binding change or the histone modification change. And if you want to understand if change in gene expression really translated to protein level, we need to perform proteomics to understand if the change at transcription translate to actual increase or decrease in protein level. Because sometimes you can have change in transcription that doesn't change anything at the protein level because you have post-transcriptional regulation that can dampen that effect. So transcriptomics, some people like this because it tells you the consequence. You have the biomarker. You know what gene changed in your disease, in your sample. But transcriptomics by itself doesn't tell you how that change occurred. You need other type of omics to combine. So we also learn about epigenomics. We know how we measure methylation or histone modification change. So this change can lead to a different chromatin state, being open or closed, active or inactive. In order to make sure that the change in histone modification really activate the chromatin, we need to check the gene expression, to check the transcription, if it really increased. And one really complex example would be related to transcription factor. So transcription factor, in general, we expect that if we have more molecules of transcription factor, it should be able to bind to the DNA and activate the transcription. But let's say you see increased binding of transcription factor to the DNA from ChIP-seq. We can still ask, does the increased binding because we have more expression of transcription factor itself? We produce more transcription factor, so more of them can bind. Or do we have more accessibility of the chromatin? The binding site opens up in certain cell type or in certain disease, and the transcription factor can access the DNA. Is that the reason? And because transcription factor sometimes doesn't act alone, they bind to DNA as a protein complex. So your transcription factor might be the same, but what changed are the other cofactors. So in order to interpret if increased transcription factor binding, what caused the increase, what the consequence of the increase, you need to combine that information with different type of omics data as well. So nothing is trivial. So you need to combine multiple line of evidence in order to interpret the mechanism. And epigenetic is popular because it links change on the genome. Change on the genome create the change in accessibility and modification, and those changes cause changes in transcription. So by having all the 3 layers, like genome tells you what change occur, transcriptome tells you the consequence of the change, epigenomics usually provide the link, the mechanistic link between these 2 cause and effect. What happens on the DNA, how does that translate to changes on the gene expression. Okay. So to give some example before we go into more details. So we saw in the single cell technology lecture that we can combine RNA sequencing and ATAC-seq. ATAC-seq measure the open accessibility of the chromatin. So people know that they can integrate ATAC-seq and RNA-seq together with the assumption that increased accessibility usually correlate with increased expression. And we can identify cell type, right, based on either, so we know that we can identify cell type based on RNA sequencing because we know the gene markers for different cell type. Right. But people have found that if you can also integrate ATAC-seq or epigenomic information, you can correlate change in RNA to change in accessibility of the chromatin. The chromatin opens up and you have more expression to the more transcription factor binding in the same area. So if you can correlate this information, you not only observe the change in gene activity, you know the cause of the increase in gene activity, which is an increase in accessibility and maybe also increase in transcription factor binding. So by combining kind of data across different layers, you have the explanation that goes along with your observation. Okay, so now we will see how the different ways that we can integrate data together. So visualizing like this is one way. Right? It's not wrong to just visualize the data and show that, see, we have more expression here and more accessibility here and more transcription factor binding here. Visualization is an okay way to integrate the data to improve your interpretation. But to be more computational, like to be more computationally sound, right? So let's see how the ways that people integrate these data. So when we think about how we integrate different type of data, right, in the single cell lecture, we actually talked about one type of data integration before, and that is the people call it a horizontal integration, but we can also integration across batches. So you have different experiments, like maybe analyzing the same disease, maybe single cell data from the same cancer, like liver cancer. So all these are different studies. They all study the same tissue, like liver tissue, and the same disease, cancer. So horizontal integration or integration across batches, we assume that different studies analyze different cells or different samples, different cohorts, but they are monitoring the same omics. This is what we are used to. We see RNA sequencing data from one lab, from another lab, from another lab. We can integrate the data by accounting for the batch effect. This is also integration, but it's integration along the same omics, a single omics. So in this lecture, we are going to focus on the other 2 types. The other one is the vertical integration. So each row here represents different omics, maybe genomics, RNA-seq, and then ATAC-seq, different omics. But they are performed on the same cells. Right? So you have a single culture, you record the DNA sample and RNA sample so that you can perform simultaneously genomics, transcriptomics, epigenomics. So vertical integration assume that you have exactly the same cells. Right? So for each cell you barcode them, right? Or for each sample you barcode it, and you analyze the same sample, right? Different record of the same sample with different omics. So there's still some kind of share information. Right? You at least you are analyzing data from the same cells. Like you know, this is the genome, this is a mutation profile from cell number 1. This is the gene expression profile from cell number 1. This is maybe the chromosome accessibility for cell number 1. Right? So you can still concatenate them together. So for example, like with vertical integration, if you observe an increase in gene expression, like for gene X, in the genomics data, you can match the change in expression for gene X to the mutation that occur around gene X. And that might explain if there is a mutation in the regulatory region near gene X. Or you might see increased copy number of gene X. And if you go to the epigenomics track, you might see change in transcription factor binding or change in chromosome accessibility. So vertical integration is still not too bad because we assume that we have the same set. But most of the time, we are in a situation that's more complicated. So we call this a diagonal integration. So this occurs, for example, like when 3 different labs are interested in the same disease, but they have different cohorts. Like so let's say a lab in Thailand perform RNA-seq on colon cancer. Right? Another lab in the US might perform epigenomic profile of colon cancer. A lab in Europe might perform proteomics on colon cancer. It's still possible to integrate the data because they are analyzing the same disease. Like we assume that at least we are monitoring the same biology. But now we have a different batches, like different cohort. And also each cohort are linked to different omics. So people want to know like if this is possible. So one of the studies try to compare vertical integration. So you have a record of the same culture versus diagonal. So you have different culture. One culture, you analyze RNA sequencing. The other culture, you analyze the chromosome accessibility. And we want to know if we have a pair, so we perform both omics, like on the same cells, like each cell we analyze both omics. Or if we have different culture and we analyze different omics on different culture, does it lead to different accuracy when we integrate the data together? So it's just to show you like the worst case, like, because most of the time you cannot go back to the same sample and extract more DNA or RNA to analyze the data. Usually if your study is done, then it's done. So most of the time in real world, we have to deal with diagonal integration. But luckily, with diagonal integration, like the assumption that we need to make. So let me see. Okay. So when we want to integrate the data like along this diagonal, the only thing that we can assume is that different cohorts, they are from the same disease. Like so, we expect like this the same or similar distribution of gene expression, similar distribution of cell type, right? Or if patient level, like we assume similar subgroup of patient. There might be different molecular signatures inside the patient. And different labs, like even though they analyze different group of patients, if the biology is the same, like the disease is the same, we should see similar expression profile, similar mutation profile across cohort. And we want to use this assumption as an anchor for integration. So let's see what this kind of comparison does. So they perform single cell RNA-seq and ATAC-seq from about 10,000 to 20,000 cells with known cell type. So this is like a well-known tissue sample where you know what cell type to expect. In this case, they have blood sample, skin sample, and they know kind of which cell types to expect. So they collect the data as a pair. So each cell, they barcode each cell and analyze both the ATAC-seq and RNA-seq. And if you notice here, so RNA-seq, like we extract RNA from the cytoplasm. But for ATAC-seq, where we investigate the accessibility of the DNA, like we don't need the RNA sample, we need the DNA, the genome. So in this case, the ATAC is performed on the nucleus. So we extract the DNA from the nucleus to perform ATAC-seq, and we extract RNA from the cytoplasm to perform RNA sequencing. So in this case, like this is a popular combination because you can isolate both RNA and DNA without interfering with each other. you isolate the nucleus, you barcode like the sample first, you isolate the nucleus, and you isolate the cytoplasm. And you can do both at the same time. Okay. So in this case, they extract the data as a pair, so they barcode each cell, analyze both omics. But to simulate diagonal integration, they shuffle the cell barcode. So even though in the original data we know which RNA-seq correspond to which ATAC-seq, they simulate the effect of diagonal by shuffling the cell barcode. So now we have no idea if which cells RNA-seq correspond to which ATAC-seq profile. So that can simulate different batches. Okay. And what they did is from 10 to 20,000 cells, like they sample 1,000 to 8,000 cells. So to mimic like a typical single cell analysis. And they want to see if they analyze the data as a pair, as vertical integration, where we know the RNA-seq and ATAC for every cell being paired, versus diagonal. Do the cell type that they infer agree with each other? We want to know if different techniques, how much information is lost when we perform the diagonal integration. And here I would like to introduce you to one of the way to compare cell type assignment between two samples. Because in this study, they know the ground truth because these are the known cell type. And they perform cell type annotation twice on vertical and diagonal integration. And they want to compare whether they are consistent or different from each other. And one of the ways that you can compare if two systems of classifying cells are agreeing or disagreeing with each other is called the adjusted RAND index. So just to show you the concept, for example, let's say we have 5 samples, like 5 cells or 5 cells. And we have the ground truth, like we know from biological knowledge that cell number 1 is a B cell, number 2 is NK cell, the rest are also B cell. But when we analyze the data, maybe from diagonal integration, we identify cell number 1, 2, 3, 4 like as the same type, and cell number 5 as a different type. So we call them A and B here because let's assume that we don't know yet from our new analysis which cell type it is, but we can cluster cells to say that these 4 are similar. We want to call them the same type, A, whatever A is, and cell number 5 as B, whatever B is. So RAND index, what they try to calculate is if you pick 2 cells, you can pick cell 1 and 2, 1 and 3, 1 and 4, 1 and 5, or 2 and 3, 2 and 4, 2 and 5. You pick a pair of cells, and you ask for the pair that you pick, let's say 1 and 3, in the ground truth, 1 and 3 are both B cells. And in your new analysis result, they are both called A. So this means that for cell 1 and 3, your result and the ground truth agree with each other because you say that cell 1 and cell 3 are the same. But if you consider cell 1 and cell 2, like sample 1 and sample 2, in the ground truth, we know that these are not the same cell, not the same cell type, B cell and NK cell. But in the integration, after integration, we call these as the same cell type. So now we know that for 1 and 2, the result disagree with the ground truth. So what RAND index does is you're going to pair up all these possible 5 choose 2 possibilities. There are 10 possibilities. And for each one, you're going to count, do they agree with between the ground truth and your result, or do they disagree? And this is called index because you're going to calculate the proportion. So out of 10 possibilities that you can pair 2 cells, 2 samples, how many times, what's the fraction, what's the proportion that the cells that you pair give you a consistent result between the 2 systems, the ground truth and your analysis. If it's high, then that means your analysis is correct. If it's low, that means your analysis disagree with the ground truth. So RAND index will go from 0 to 1. with one being that your result perfectly agree with the ground truth. And you might ask, like, why don't we calculate like something like an accuracy? Like, how many out of 4 B cells, like how many can we call as B cells in the new result? So people use RAND index instead of something like accuracy because sometimes when you cluster the cells like using your omics data, if you use RNA sequencing, then you can identify them as B cell or NK cell like by looking at the markers for B cell and markers for NK cell. But if you cluster it using ATAC-seq, if you cluster them using similarity in chromatin accessibility, or if you cluster the cells based on their mutation profile, then you have no idea what they are. Like you can call these A and B, but because you don't have transcriptomics, like if you don't have transcriptomics, you can never tell that whether A is B cell because you don't know the marker, the expression of the B cell marker. So RAND index is more generalized because it allows you to compare two systems that use different names. You can have cell type here and just characters here, and you can still calculate how they agree with each other. So that's why they use RAND index for this purpose. So in this example, if you count out of 10 possibilities, you have four pairs that agree between ground truth and the new result. And those are the pair between 1, 3, and 4. They are both from A and from B cell. And 2 and 5, because they disagree, they are not from the same cell type. Okay. So this is a metric that they use to understand if after vertical or diagonal integration, do they still get the result that's agree with the known cell type. So let's look at their result. So in this case, they are showing you 3 different analyses. So they collect the data as pair. So for each cell, we know the RNA-seq and the ATAC-seq. That's the blue color here. These are performance, the agreement that you get if you use the information that the same cell, you know both RNA-seq and the ATAC-seq. So this is a perfect condition. For the yellow, it became unpaired, so they shuffle the cell barcode. So now you don't know which RNA-seq corresponds to which ATAC-seq. This is the yellow condition. But yellow condition compared to the green condition, there is also one more difference. So in the yellow condition, they collect the same set of cells, like they analyze RNA-seq and ATAC-seq on the same set of cells. But they shuffle the barcode and get the yellow condition. It's unpaired now, but the data actually come from the same population, the exact same cell. But in real life, when you perform diagonal integration, cells that you have the RNA-seq data for might come from one lab, and cells that you have the ATAC data for come from a different lab. So it's not the same cell population. So yellow here is still, there's a little bit of cheating inside because they assume that you get the exact same cells in the experiment. But in real life, it should be the green condition where the cells that you analyze are different for each omics. And what you can see here is there is only a slight change in the adjusted RAND index. So 1 is the best, 0 is the worst. So there's only a small difference, small performance difference. Even if you do like a pure diagonal integration, data from different labs unrelated to each other. Versus if you have the exact same cells, but you don't have a matched RNA-seq and ATAC-seq, and the perfect condition where you have the vertical integration. The agreement change very little. But one thing that is interesting, I don't include the full information here, but different colors correspond to different bioinformatic tools. So even though you don't lose a lot of kind of analysis power by going from diagonal to vertical integration, but the choice of tools matters a lot. If you pick the wrong tools, like the tools that perform well for the vertical alignment, somehow doesn't perform well for the diagonal. And tools that perform well for the diagonal alignment perform poorly for when you have the actual vertical data. So the choice of tools is important because these tools have different assumptions. The tools in blue here, this one, it performs best in the green condition, the diagonal integration, because it assumes that your data is diagonal. It doesn't assume that you have data from the same cells. It assumes that your data comes from different batches, different laboratories. But the yellow tools here, the dark yellow line here, these tools assume that your data is paired, and it uses that information to call the cell type. So it performs best when you have the actual pair data. So in practice, like, you don't have to worry too much when you integrate data across laboratory, because with the right assumption and the right tools, you can still recover similar cell type classification performance. And in the same study, like, they show you also how the ground truth cell type correspond to the prediction using different scenarios. So blue again, so blue is the perfect vertical integration. Yellow is the simulated diagonal integration. So if you look carefully, like where I put the arrow, none of the method and even the perfectly paired data, right, doesn't give you the perfect cell type classification, cell type annotation. So the issue of kind of having batch effect across laboratory is not that big of a problem, because the actual variability in your data is kind of stronger. So you don't have to worry about the fact that you have different labs perform different omics. So at least we don't have to worry about that. So this is like one viewpoint that we can see how we integrate different type of omics data under different scenario. And at least from this paper, at least for the purpose of cell type classification, we don't see too much difference between the scenario. So that's good news. So next, let's look at how we can actually combine the data to make interpretation. So the diagram that we saw before, like horizontal, vertical, diagonal, those are ways to handle batch effect. But once you have multi-omics data, how do you combine them together to make a decision? So there are also 3 different ways. There are probably more ways, but 3 major ways that you can imagine how multi-omics data can be used. So for the first way, this is the diagonal integration. So you have different type of omics from different laboratories, and we want to aggregate them first, like using diagonal techniques, and then maybe call the cell type. So we combine data from different omics, assume that they describe the same disease, the same biological system, and then analyze the data. So this is a common approach in single cell data. We combine different single cell omics and then make the interpretation together. What we will see next week will be the second technique here, where we want to transform each omics data onto a common structure. We will talk more details in a few slides. So instead of kind of concatenating them diagonally, we want to transform them first. Sometimes people call this like a transformative integration. So we want to transform the data in some form, and that form would be the network. And we want to integrate the agreement between the networks to create a single network to interpret the data. We will look into more detail in future. And the last one, this is also possible. You can use each omics data independently. You can use genomics data to identify some biomarker. You can use transcriptomic data to identify some biomarker. You can use proteomics data or epigenomics data to identify some biomarkers. And then you can intersect these biomarkers together to identify genes, mutation, and protein that works together. Okay, so let's look at them one by one. So the first one, this is the concatenation or diagonal integration. This is what people usually use for single cell omics. So we assume that different omics come from the same biological system. They are not exactly the same cell, not exactly the same cohort, but the same disease, the same tissue. So they have the same distribution. So we already saw this in the single cell lecture. We can integrate RNA sequencing with ATAC-seq by using this assumption. So we have seen this before. This is nothing new. It's also multi-omics, but we have seen this before. The second one I will read for the, okay, I think I switched the order a little bit, so based on complexity. So the third technique that I talked before, you can analyze different omics separately. Like for example, you might identify differential gene expression from your transcriptome. You might identify differential protein from proteomics. And you also identify different transcription factor binding, methylation or histone modification from ChIP-seq. And you may identify change in mutation from genomics. You might identify change in metabolite from metabolomics. And what we want to do is, once we identify all these statistically different changes, like in different omics, we want to ask, do they share anything? For example, do we identify the same gene being differentially expressed and the same protein being differentially expressed? Gene translate into protein. And do we see transcription factor binding changes near the genes or the protein that have differential expression? We want to see agreement across the layers without actually combining them in the analysis. When we perform differential gene expression, we only analyze transcriptomics. When we perform differential protein expression, we only analyze proteomics. So we analyze the data separately, but we can still combine them afterward by seeing if they identify the gene, they identify the area of the genome, they identify the metabolite, do they correspond to each other? Are they in the same pathways? Are they regulating each other in some way? This is also a way to integrate the data very simply. The technique that we will talk more next week, but I will introduce a little bit now. So sometimes when you want to integrate omics data, so in the case of concatenation, we want to integrate the omics by focusing on the cells, on the sample. We want to show that these cells, these samples have RNA sequencing and ATAC-seq that correspond to each other. We want to integrate cells and sample. For this type of independent modeling approach, you don't integrate the cells. Right? In transcriptomics, you analyze the sample, whatever cells they are, whatever sample they are. In the epigenetics, you analyze the sample, might be from different cells, but from the same disease. You don't care about whether they're the same cells. But what you want to see is, if I'm analyzing the same disease, like the same tissue, do I see the same biomarkers in different omics layers? Do I see the same biomarker? So the focus can change from integrating the cells to integrating the biomarkers. And the last one here is integrating the similarity between cells or between tissue, between cells or between samples. So let's imagine if you have a cohort of patients, and in this cohort, you analyze different omics data. You identify mutation from genomics, you look at differential expression from transcriptomics, you identify chromatin accessibility from ATAC-seq from the same group of patients or for the same group of sample. So what you want to create here is a network, and you want to connect patients that have similar omics profile. So based on gene expression, you can create one network. So this line connects two patients that have the same or similar gene expression. This line connects these two patients that have similar gene expression. So you have one network based on gene expression. But if you analyze genomics, like you look at the mutation profile, you might see a different structure. These two patients might have similar mutation. These two have similar mutation. These two have similar mutation. And they might be different from the similarity in gene expression. You have this similarity in gene expression, but not in mutation. So from different omics, you can draw different connectivities between patients, between samples. But because we want to be able to say that patients are similar as a whole, right? Not based on just one omic. So the purpose of this technique is to integrate these three networks of patient-patient similarity into a single network of patient-patient similarity that capture the similarity across all omics. So for example, you might want to say that these two patients are similar because they are similar in 2 out of 3 omics. So the majority say that these two patients are probably similar, but somehow the mutations are different. But they might have a different mutation, but still the same effect on gene expression. That's possible. So disagreement between omics can occur, but we want to make interpretation as a whole. Based on all 3 omics that I see, which patients are similar, which patients are not similar. So sometimes people call this technique a network fusion. So we create a network of patient-patient similarity, and we want to fuse, we want to merge multiple networks together. So in one example, this is from a study from more than 10 years ago. So they analyzed for the same cancer patient, the same cohort. They analyzed gene expression, DNA methylation profile, and also microarray expression. And from gene expression, you can have one picture of patient-patient similarity. So these 3 patients have similar DNA, similar gene expression. These 3 patients have similar gene expression. These patients have different gene expression. But from the perspective of DNA methylation, somehow these 3 patients have similar DNA methylation profile. These 3 patients have similar DNA methylation profile, but these 3 patients do not have similar gene expression, do not have DNA methylation profile, similar DNA methylation profile. So you have some agreement between transcriptomic and DNA methylation, and some disagreement. So the purpose of network fusion is to merge these 2 pictures of patient-patient similarity to create a single structure that will allow us to interpret patients as different subpopulations. So these 3 are patients with similar DNA methylation and agreement between DNA methylation and gene expression. These are the 3 patients with most of the similarity being based on transcriptomic, but also there is some evidence from DNA methylation. And these 3 patients are similar in both aspects. So we want to use different omics to cluster the patient, to group the patient or group the samples together. And because in the beginning, different omics give us a different picture of patient-patient similarity or sample-sample similarity, we want some kind of algorithms that can average out or aggregate or merge the connectivities between these 2 networks until we arrive at something that is an overlap between the 2. So let's look at a little bit like the idea behind this network fusion algorithms. So let's say we start with gene expression. We start with this patient-patient similarity. And what we want to do with network fusion is we want to make changes to the patient-patient similarity based on gene expression to make it more in agreement with DNA methylation. So basically we want to change the blue graph here to make it more similar to the purple graph, to the pink graph below. So we need to make some changes to the edges, to the score on the edges, somehow to make this graph more similar to this. So the idea is this. So we start with the gene expression similarity network. So we have the blue graph, this blue graph. And we focus on 2 patients, patient I and patient J. We want to make changes to I and J, between I and J. So if we pick these 2 patients, based on gene expression, they are not similar at all. But based on DNA methylation, they are very similar. So we want to introduce this similarity into this network, into these missing edges. somehow. So the idea is this. Because these two patients, they are similar in one omics, but not similar in the other. So we don't have ideas about which one to trust. We don't know if we should keep this because we trust gene expression, or should we remove this because we trust DNA methylation. So instead of focusing on just a single connection between patient I and J, this network fusion algorithm will look at connection between I and J through other patients. So let's say patient I has a similar gene expression profile with patient K, another patient with similar gene expression profile. And patient J has similar expression profile to patient L, a different patient. Because I and K are similar and J and L are similar, so if we want to update, we want to make changes to our belief, to our information about I and J, what we can do is we can ask, since K is similar to I, K may be a stand-in for I. L is similar to J, so L may act as a stand-in for J. So we are going to ask based on DNA methylation, we switch to a different omics. Based on DNA methylation, if I keep changing K, I pick patient K that are similar to I, that can be multiple patients, and L, there can also be multiple patients that are similar to J. If I pick K and L among this group, among this population, and I ask, are K and L similar based on DNA methylation? So the orange edges here is based on DNA methylation. If K and L, whichever K and L I pick, they are similar based on DNA methylation, I want to say that, oh, maybe I and J is not that, is also similar, can also be similar in terms of methylation as well, because other patients that are similar to them have similar DNA methylation profile. So I can infer that maybe I and J can have some DNA methylation similarity in that. Even if in the original data, I don't see the similarity, but because their friends, like other patients, are so similar, maybe these two are also, we can also say that these two are similar as well. But if I change patient K and L, I pick among the other patient, and K and L don't have similar DNA methylation, then I'm going to say that, then I and J probably don't have similar DNA methylation. So the network fusion algorithm's idea is to infer the similarity between I and J for the different omics by looking at the omics data on the neighbors or on the friends of I and J, and use that information to stand in for the connectivity between I and J. So hopefully that kind of shows you the concept. So basically, so let's focus on these three patients as an example. So based on DNA expression, based on gene expression, they are not similar. But when we look at their friends, they are somewhat similar based on DNA methylation. So we can make inference that maybe they are actually similar, but when we collect the data, we don't see that similarity at all. But because we see a strong similarity among their neighbors in a different omics, we make the inference that maybe these three are similar in some way, but it reflects across a different omics instead of across gene expression. If we focus on these three patients are similar both on gene expression and on DNA methylation. So in this case, it's easy, right? We connect these three together because they are similar in both omics. For the three patients in the bottom here, they are similar in gene expression, but not similar with DNA methylation. But because we see this kind of many edges connecting neighbors together, we can infer that maybe these three kind of form a group of similar patients. But that similarity is more reflected in gene expression and less in DNA methylation. So this is the result from the analysis. So they have a group of patients, and they have 3 omics: gene expression, DNA methylation, and microRNA. And from different omics, they have different similarity. So these 3 graphs are not the same. But by clustering based on similarity, not between each patient pair, but between their neighbors, they can identify that there are 3 groups of patients. This group of patients, some of them are similar based on gene expression, some of them are similar based on DNA methylation, some of them are similar based on microRNA. But overall, you can see that there are many connectivities, many edges among these patients. The same for this cluster here. We see a lot of similarity in gene expression in only some part of the groups. And we see similarity in DNA methylation in a different part of the group. And we see similarity in microRNA for some part of the group. But these 3, you can see that when overlap on each other, you have similarity that not only connect between two patients, but connect between groups of patients together. So with network fusion, we can kind of reconcile, we can merge similarity from different omics to identify a group of patients or a group of samples that share similarity across multiple omics data types. So another technique that we can use to integrate omics data use a term called latent factor. So I want to introduce this term first. You might have heard the term latent in statistics class before. Actually, in this course, we talked about latent factor, but we didn't use the term latent factor. So when we learn about hidden Markov model, if you remember, so hidden Markov model, so in this example, this is a hidden Markov model for intron and exon DNA sequence. So when you are in exon mode, you have a certain property of nucleotide. Exon might have no bias. Exon can be any nucleotide, A, T, C, and G, with the same probability. And exon can transform into the splice site to go into intron, maybe with certain probability. Most of the time, you will generate a long exon before you go into the splice site. In the splice site, there's a bias toward G and sometimes A. So you have different nucleotide frequency for the 5' splice site. And the intron, let's say we have a bias toward A and T, like 0.4 and 0.4, and less frequency of C and G, so low CG content. So in hidden Markov model, we call these exon 5' splice site and intron state, like the hidden state of the model. The idea of hidden Markov model is when we observe the data, we observe the nucleotide, we sequence the nucleotide. We don't know the state. We don't know if this nucleotide is exon or intron. But we want to make prediction. We want to make inference whether this sequence, which part corresponds to the exon, which part corresponds to the intron, and which part is my 5' splice site. In statistics, you can call these states, these hidden states, as latent factor. So latent factor, if we have to define, latent factor are what we use to describe characteristics, interpretable characteristics that produce the observation. So in this case, we call the hidden state latent factor because if you know the state, like if you know that it's exon at this position, you know the frequency of nucleotide. Basically, if you know the latent factor, you know what to expect in your observation. But when we do experiment, when we collect data, we have the observation, but we don't know the state. Like we want to make inference about the state. So most of the time, latent factor is unknown. It's something that you have to use statistics or data analysis to infer or to predict. But the useful things about latent factor is that if you can assume that you know the latent factor, you can generate the observation. So if you know that these are exon, 5' and then intron, you can generate a gene sequence because you know the frequency for nucleotide, for exon position, for 5' position, for intron position. So for example, if we know the cell type, you can have expectation about the gene expression because you know different cell type have a different gene expression profile and different markers. If you know the disease state of the patient or the animal, you know that this is healthy, this is disease. You know how you know what to expect for the phenotype from your measurement. So in statistical model, we also make use of this latent factor, but in a hidden way. So when we correct for the batch effect, when we cluster cells into different types, the batch ID and the cluster ID, these are also latent factor because if we know them, we can model the effect. If we know which batch each sample corresponds to, we can calculate the batch effect for each batch. If we don't know, then we have no idea how to calculate the batch effect. If we cluster cells into, if you group cells into clusters, and we know the cluster ID, we have some expectation that each cluster should have cell in the same cluster should have similar gene expression profile. And we can, because, so these two are different in the sense that the batches you usually know beforehand, when you analyze the data, when you acquire the data, you know that this is batch number 1, this is batch number 2, this is batch number 3. But when you cluster the cells into cell type, in the beginning, you don't actually know the answer. In the beginning, you don't know which cell is B cell, which cell is T cell, which cell is NK cell. But if you know, if someone gives you the label, like this cell is B cell, this cell is NK cell, this cell is T cell, you can tell if it's correct or not because you know the marker for B cell, for T cell, for NK cell. So in essence, like some statistical techniques will keep optimizing the latent variable until the latent factor match the data. So for cell type, like you can imagine that if you have a single cell data and you know that in here there are B cell and T cell, but you don't know which is which. If you are allowed to guess, like you guess that cell number 1 I think is B cell, cell number 2 maybe it's T cell, cell number 3 is B cell. If you are allowed to guess, once you make a guess, you can evaluate whether your B cell express B cell marker, whether your T cell express your T cell marker. And if you are allowed to change your guess, you can keep changing your B cell T cell assignment until your assignment perfectly fit the known markers for B cell and T cell. So in a sense, like latent variable can be used as a proxy to optimize the assignment of cells into cell type, patients into groups, for example. So how does this translate to multi-omics analysis? So when we want to combine like, different omics data together, one information that we assume that are shared, like even though these are from different labs, maybe different cohorts, one information that we assume that are shared across these omics data is that they capture the same biology, the same disease. So some people want to model this explicitly. What do we mean by we assume that these data come from the same biology? So what they want to do is called a factor analysis or decomposition analysis. So what they want to say is that if you have different omics data for the same sample, so let's say this is a vertical integration, you have the same sample and you analyze the same sample in different way, in different omics. Because they are from the same sample, maybe patient number 1 have a certain disease. Okay. So each patient or each sample can have different latent factor. The first patient might be having one disease, taking certain drugs, come from different population, for example. This can be the latent factor for the patient. And if we assume that these latent factors like population, race, age, drug that they are taking, disease state, if we assume that these factors determine the omics result, if the patient is from a certain population, we might expect certain mutation. If the patient has a disease, we might expect a certain gene expression profile. So the description of the patient can determine the omics data that you are seeing. So in terms of statistics, we want to write out, not sure if you are familiar with this type of decomposition. If you recall metrics, in omics data, we have a sample and feature being the two dimensions of the data. Samples, cells, and then features like genes, expression, mutation, for example. We have samples and measurement. Feature here mean measurement. We want to decompose sample by feature metrics into two metrics that multiply with each other. The first one is the measurement and factors. The other one is factor by sample. So if you recall matrix multiplication, if you multiply a matrix with features times factor to the matrix with factors times sample, the result will give you a matrix with feature times sample. So in this case, they want to use the latent factor like patient age, disease state, population, race, for example, as the factor that combine. So in the factor by sample table, so this is a descriptor for your sample, cell type, dose, drug dosage, for example. But in the feature times factor metrics, these are the ones that you don't know. For your sample, for your patient, and you can collect the metadata. So you can generate this metric, factor by sample. But what statistical model wants to learn is, can we discover how different factors result in different omics measurement? Can we reconstruct these metrics, these three metrics, so that when we multiply each of them to our metadata, we can reproduce our omics data? If we can do this, then that means we can explain multi-omics for the same sample population based on their metadata. And if we can do this, similar to when we mentioned about PCA last time, if we can do this, then we can ask, for example, we might have 10 factors here, one being age, being drug dosage, being disease. We can ask, is factor number 1 good for explaining drug response data? Is factor number 1 good for explaining DNA maturation profile? Is factor number 1 good for explaining DNA mutation profile? Because these are just numbers, so we can look at the coefficient here to see which factors has a strong coefficient against which omics measurement. So we can analyze the association between each factor. So which each column in your metadata against each omics type. So now from this result, we can tell that whichever factor 1 is, might be some description about the patient or the cell, somehow it's correlating with DNA maturation. It's correlating with mutation. We can also say that for mutation, looks like it's being explained by only 2 factors. These are not important. For DNA, for gene expression, you require many factors to explain, because DNA gene expression is more complex. In your data, you might have a few causative mutations, so it's only correlated with certain factors. But to explain gene expression, you need a lot of information from your metadata. Different age give a different gene expression. Different drug dosage give a different gene expression. And in this case, DNA maturation profile seems to be regulated by only one factor. So these are things that we can interpret if we are able to make this decomposition. We can interpret the coefficients that we learn from the data. So just to recap, metrics on the left, these are omics measurements, these are data that you get. You don't have to make any guess about these. These are the data that you collect. These are your metadata table for your sample. These are things that you collect as well. What the statistical model does is to infer these coefficients in the middle here that link your factor in your metadata to the omics measurement, each omics measurement. Okay. So overall, we can see which factor correlate with each omics type. But because we actually have coefficient for every measurement, so we actually know the association between each factor versus every gene expression versus every mutation versus every DNA methylation. So you can even go into further detail and ask which mutation on which gene that are associated with factor 1. And in this study, they found IGHV mutation and trisomy on chromosome 12 to be related to factor 1 and factor 2 respectively. So this allows you to combine multiple omics data type and make association analysis about which metadata, which descriptor of your sample are linked to each omics measurement. So this is for mutation. But if we also ask for factor 1, factor 1 may be associated with different gene expression, different mutation. If we ask so what are linked to factor 1 regardless of omics? On the mutation, we already found this gene, this mutation of IGHV being linked to factor 1. On gene expression, we might find increased expression of this gene being linked to factor 1. And in this case, many of these genes are known to interact with IGHV. So we see some consistency between affected genes in terms of mutation versus affected gene expression that are related to the same gene. And if we link this to, let's say, drug treatment or other type of omics, we can also link findings from gene expression and mutation to also drug response. So this is another way that you can combine different omics data types. Okay. So just to summarize, so we talked about different ways that we have multiple type of omics. We want to combine them. We have the motivation. Different omics provide different biological information. If we can combine them, we can make a conclusion about the mechanisms. And we saw in one example of the study that we can combine event data from different labs, different cell culture, different population cohort with minimal problem. So that's good news. We can integrate data. But the key point is we need to pick the right tool. We need to pick the right strategy to integrate the data because different techniques are good for different situations. So in this case, before you analyze the data, you probably have to read some review article to identify which techniques or which tools are good for which situation, and you can follow similar analysis. Okay, so that's everything I have for today. So next week, we're going to close out the system biology module with the topic on biological networks, where we draw the graph, we draw the networks that connect patients, connect cells, for example. Okay.